Okay, today's lecture is going to be on sterilization, disinfection, and antibiotic therapy. Looking at this image to start with, this is something we've seen a little bit about before. So the background cream colored on these plates that we see, that's bacteria. And what we see here, these clearings are area the bacteria can't grow. So this is something we've seen similar with the plaque assay that breaks up and lyses open cells. And now we're going to see antibiotics and how they can do the same thing with bacteria and how we can use this method to test if antibiotics are effective or not. Okay. Looking at this section, we're going to be looking at different modes of action. For antimicrobial agents. We're going to be looking at everything from sterilization to disinfectants. We're going to learn the difference between that vocabulary. Finally, we're going to be looking at clinically effective ways of doing things. So if we are in the allied health, this would be an important topic. We'll also be looking at the empirical theory, which is when someone goes into the emergency room. Typically, antibiotics are prescribed based on the signs and symptoms, so based off the syndrome that's seen. It's kind of the first line of defense. And looking into antibiotics, diving a little bit deeper, we're going to be looking at selective toxicity with them, how antibiotics essentially work by killing microorganisms, but don't harm our cells directly. And finally, we'll look at antibiotic resistance. So we'll look at the modes that antibiotics work, and then we'll look at the modes of how microorganisms resist antibiotics. Okay, looking at microbial measures, we have sterilization, disinfection, antisepsis, and sanitation. Looking at all of these, we can see from the top down we see that they are stricter to less strict. So sterilization is destruction of all living cells, but it doesn't only contain living organisms. We also see that this is relevant to viruses as well. So this is the strictest form of antimicrobial measures, followed by disinfection. So disinfection is removing any disease-causing microorganisms from inanimate objects. It's an important distinction there. Because when we look at antiseptics, here we're removing disease-causing microorganisms, but on living tissue. So in a clinical setting, we could see a disinfectant used on maybe a hospital bed or tools, where an antiseptic is used maybe on a wound or living tissue. And sterilization would be something that's done on maybe surgical tools that are going to be used. And looking at sanitation, this is probably, we've heard the term sanitation before. This is just actually reducing the amount of microorganisms because it's not only if they're pathogenic, it's also the number of microorganisms is going to depend whether an infection can be mounted or not. So we want to reduce them to a safe level. So sterilization itself is destruction of all living and non-living for viruses. Everything below that is less stringent when it comes to its antimicrobial measure. Okay, when we look at antimicrobials, we can break them into two categories. We have cytal agents and static agents. Cytal agents are the ones that actually kill the organism. So something that's bacterial cytal is going to kill bacteria. We can also have fungicides, virucides, algicides. As you can imagine, fungicides kills fungus, virucides kills viruses, and algicides kills algae. So these are all going to kill the microorganism, essentially, even though viruses are non-living. Then we move on to our static agents. Static agents are a little bit different. They hinder the progress of the microorganism. So this could be by slowing down the metabolism. Metabolism can also play into reproduction, as in binary fission of cells reproducing and replicating. So we are slowing down the infection rate. So we call these drugs static. So we could apply that again, the same terminology, bacterial static, fungus static, virus static, and algae static, meaning it's slowing the progress of these organisms or viruses, essentially. You might be wondering, why would we want to use a why would we want to use a cytal agent versus a static agent? It seems like cytal would be the go-to. Well, static agents are also helpful, and sometimes we use them in combinations. So a static agent can help control an infection. We talk about sanitation here and reducing microbes to safe levels. Well, the amount of microorganisms that we have can change whether they can mount an infection or not. So by using a static agent, we can actually control the growth of the microorganisms and potentially let our own immune system work to clear out the infection. So it can work mutually with our immune system. Okay. Looking at sterilization and cytal agents, we can look at how they actually work, but there's going to be a change in what we're treating. So we have to look at the target organism. So population size is going to be a big thing. Do we have microorganisms that have been just left to grow over a long period of time, leaving us billions and trillions of cells to deal with, or is this a small infection that we're dealing with? So this is going to change the cytal agent, or this is going to change the static agent that we use and the dosage of it as well. Type of cells in a population is also important. If we have spores, which are like endospores, we know that a lot of methods like disinfection is not going to remove spores. So spores are only removed, if we go back to our first slide here, with sterilization. Everything following sterilization does not kill spores. So that's a good distinction to make. Okay. If we look at the duration of the exposure to the lethal agent and agent concentration, that's going to be really important. So we could use something like a disinfectant, spray down a surface and wipe it off directly. That's not going to disinfect the surface. So this is a common misunderstanding. Just because you're using an agent at the proper concentration, the time of exposure is actually going to be really, really important too. If you're in the allied health science, this is something to pay attention to, because that's how we can disinfect appropriately. The amount of time that the agent has to really work on the microorganism is going to be important. And following that, we have organic load. So if we're looking at something like blood or feces, what kind of medium is it in? If we just spray it with a disinfectant, are we actually going to remove the microorganisms? Well, possibly not. If we have something, a matrix that's suspending the microorganisms, like a fecal specimen, well, if we put an agent on top of it, it's not going to get into and penetrate through the organic material in order to kill the microorganisms inside. So organic load is important as well, and it's going to change again the type of reagent that we're going to use and the duration of exposure. Let's look at sterilizing and cytal agents and how we can use them to kill exponentially. We use a measure called the D value, which is when we can kill 90% of the cells. Looking at this graph, on the x-axis, we have exposure time. On the y-axis, we have viable organisms. So here we can see this test tube with all of the organisms in green. Here, the viable organisms, we're at 100%. We have all the organisms living there. Then, as we move down here, we see that 90% of them are red, indicating that they are no longer alive, and only one that's green. So here we've killed 90% of the cells. So this is essentially our D value that we're looking at, going from 100 to 90 here. And that's our exposure time. So now we know how long we need to expose for in order to kill 90% of the organism. So almost 5 minutes here. Okay, let's look at methods of sterilization, decontamination. As we move through these, we're going to see some that are more common than others. So here we can see an autoclave. An autoclave is a very common way of sterilizing laboratory equipment and also sterilizing equipment used in surgeries. So any of the metal surgical tools are typically put in an autoclave. We can even see this at a dental office. They have smaller autoclaves in which they add their equipment into these little blue and white bags, and they place them in the autoclave. Those little sacs allow for steam to penetrate inside the bag, And there's actually a chemical change, a color change that you can observe to be sure that the actual autoclave worked and that it heated up to a standard temperature. The autoclave works by steam and pressure. So it heats up to 121 degrees Celsius at 15 psi. So that's the pressure. And it runs for approximately 20 minutes or more. 20 minutes is the standard, though. During this procedure, this steam is actually sterilizing everything within the autoclave. So again, most laboratory equipment is cleaned through an autoclave so it can be reused again. And surgical equipment is cleaned this way as well. So they're placed into the autoclave and it's ran for approximately 20 minutes and steam sterilized. Let's look at the 12D method. The 12D method is another way to sterilize. So these are, again, are more concentrated ways of killing microorganism. 12D can kill up to 10 to the 12th endospores. So endospores are really, really difficult to kill. So autoclaves and endospores can do this. So this is why we call it sterilization, because it's killing a good amount of endospores here. And if this method continued, you could potentially kill all of the endospores. So remember, endospores are a metabolically inactive state of cells, so kind of like a dormant state, and they build this cortex around them that protects them from the outside environment. Microorganisms can live in an endospore form for thousands to millions of years. And they're just fine because they're not metabolically active. Then they can turn back into vegetative cells and be quite dangerous. So a lot of our Clostridium species are going to be the ones that are forming endospores. So we have Clostridium botulinum, which can be extremely dangerous and deadly, especially to infants. So canning is a great way to keep us free of Clostridium botulinum by killing off endospores with the 12D method. So this is our canned food method, and this is why canned food lasts for so long. Okay. Looking at pasteurization, so pasteurization is using heat to kill microorganisms. This was invented by Louis Pasteur, as you could imagine, in 1864. What it does is it brings food up to a certain temperature to kill off a good percent of the bacteria. So we're not killing off all the bacteria. This is not sterilization, but it helps prevent food from spoiling, so it can have a longer shelf life. Um, So it's going to kill off, again, microorganisms. So this can prevent microorganisms from fermenting inside of foods. If we've ever had something that's maybe been in the fridge for too long or left out for too long and it has kind of a funny alcohol smell, that's because the microorganisms within it have actually started fermenting. So our nose is a great way to determine if something has been spoiled, if something goes wrong and we shouldn't be eating it, because those microorganisms are continuously replicating and maybe making these kind of odd alcohol fermentation products that we can smell. Okay. It says here that pasteurization doesn't affect temperature or doesn't affect texture, color, or taste. Many people actually disagree. So we pasteurize a lot of things here in the US. We pasteurize orange juice, we pasteurize milk. But a lot of people who drink raw orange juice or raw milk will say, yes, it does actually affect the flavor profile of these products. But pasteurization has saved many, many lives. Especially children's lives whose immune system is not as strong as an adult. So if there's any kind of bacteria within the product that we're giving them, high percents of it, pasteurization will prevent an infection from taking place. Before pasteurization, many children died from drinking raw products like raw milk and raw orange juice. A common way that we use at home, or we can even use in a lab too, to control the growth of microorganisms is going to be refrigeration. Refrigeration works by not being the ideal condition for microorganisms to grow in. So most organisms that are pathogenic to us are what we call mesophilic. They live in the range of our body temperature, and that's their ideal condition. Once we go too cold or too hot, it's no longer ideal for these microorganisms to live. So by putting our food in the refrigerator, it prevents microorganisms from growing. So they stop at where they are, the amount of microorganisms there are on the food at that point, or go very slowly from there on out. Before refrigeration, we actually used to salt foods. So salting foods is a way of using an osmotic difference to cause bacteria not to be able to grow, to not be, again, an optimal condition for them. Because most microorganisms that are pathogenic are not halophiles. They're not interested in living in a high salt concentration. Putting salt on food causes water to be purged from it. As the water is purged from it, it's also purged from the microorganisms, so they can die from desiccation, from drying out. So we used to salt our foods. Now we do refrigeration. We know if we leave something in the refrigerator for long enough, microorganisms will continue to grow. It's not as good as freezing our foods. So they're still metabolically active. They're just not in a highly metabolically active state. There's one exception to point out here with refrigeration that's of interest. It's listeria. Listeria, we always see common outbreaks with, and it's typically in dairy products. So listeria has no problem growing at the temperature range of the fridge. So if you take some, let's say a yogurt, and you put it into the fridge, and it does have listeria in it, if it's contaminated with that microorganism, the fridge will not prevent it from growing. So it will continue to rapidly grow. And this is why we always have listeria outbreaks, because our modes of controlling bacterial growth don't necessarily work with refrigeration against listeria. If you want to check out the cdc.org website, you're going to see the listeria pops up quite a bit on there. Okay, let's look at freezing. So we know that if we have something in the fridge, microorganisms will grow slowly, but if we put it into the freezer, they'll essentially almost stop growing. So again, changing the temperature to be outside the optimal condition of the microorganisms is going to prevent them from growing. So we can store things in the refrigerator for a couple days, in the freezer, sometimes up to years, because we're preventing microbial growth of our food. So freezing can also be done in the laboratory. We mix glycerol with our samples. This way that the ice crystals in freezing doesn't rip through the tissue of the microorganisms. So as we go from refrigeration to freezing, water crystallizes. Crystallization can tear apart cells, like little daggers that move into them and lice them open. But we mix glycerol in with our samples because glycerol keeps a viscous solution, so we don't form those ice crystals that penetrate through the cells. So 1-to-1 with glycerol is a great way to keep your sample in the freezer, so you can culture your microorganisms and freeze them. Okay, we also have lyophilization, which is what we know is freeze drying. It's done in a vacuum, and it's a very quick way to freeze products. So normally when you take something and you place it into the freezer, it takes a couple hours before it's frozen. Freeze drying, on the other hand, is quite almost instant here in comparison. Okay, let's look at filtration. Another thing to mention, freeze drying. That's what's done a lot with commercial food products that we buy that are frozen. So they've been lyophilized, so they've been instantly frozen, and instantly freezes them, prevents microorganisms from continuously growing while they're going through the process of freezing. Remember, some of them can divide every 20 minutes or less. So if we can freeze something quicker, we have less microbial growth on it. Let's look at filtration as a means of removing microorganisms. So what we see here is a syringe with bacteria in it. Then we see a filter inside of here, and then we see the solution outside of here. So what we can do is we can syringe filter microorganisms. The filter that we have here has a pore size of 0.2 micron, and we see this is the filter zoomed in on. We can see the microorganisms resting on this filter. It's really a cellulose filter, and they can't go through the filter, so we end up with a sterile solution. Depending on the size of the filter, we'll remove different things. So 0.2 microns can remove endospores, yeast, mold, algae, and of course protozoa that are very large eukaryotic cells. But we can capture them on the filter, and that's how we can get sterilized water. Let's see. So I think we've been seeing things like straws like this, that a straw can be used with a filter in it that prevents the microorganisms from being brought in. So the microorganisms will build up on the filter. They're caught by the filter, essentially. If we want to remove viruses, we would need a filter with even smaller pore sizes. So 20 nanometers here would would prevent even viruses from moving through. So when we first were learning about viruses from bacteria, we learned that they were so small that they couldn't be picked up in the common filtration methods. So we had to advance filtration to even smaller pore sizes to capture them. My work in aeromicrobiology is capturing microorganisms within the stratosphere. I actually capture them on filters like this. So we have airflow, passive airflow over the filters on a scent of my bioaerosol sampler. And I have a small micron, it's actually 0.45, which is sufficient for what I'm doing to capture most of the microorganisms. And then I can bring them back down and sample them and find out more about them based off of their DNA. Okay. Irradiation is another way to kill microorganisms. So to kill microorganisms, we see that they've been approved in the United States from the 60s up until 2010 is where this book goes up to. Different ways of irradiation is like using UV light, gamma rays, electron rays, and X-rays. And this kills microorganisms on the food before it goes to market. So here we can see for fruits and veggies, it can control insects. For pork, it can control parasites that we can find in the muscle tissue of pork. We can see we can control mold in wheat flour. So lettuce and spinach, again, reducing bacterial pathogens. So all of this irradiation is removing microorganisms from food. Now, remember, we've typically eaten food naturally that has microorganisms on it. There's nothing wrong with that. But if we want to remove pathogenic microorganisms, we can take on something like irradiation to kill off almost all microorganisms on the food. So that would remove the pathogenic ones as well. Okay, let's talk about disinfectants. So again, disinfectants are not going to be used on the body. Disinfectants are used to sterilize the environment around us. Disinfectants are a great way to remove fomites from, let's say, doorknobs or common things that people touch. So we're going to go into the use dilution test here, which can tell us a little bit more about the phenol use dilution and phenol coefficient test to see how disinfectants work. So here we can see an image of microorganisms together, and so we'll see how it can work to kill them. Moving through disinfectants, actually, killing microorganisms, whether we're using use dilution or the phenol coefficient test, it's really just a way to rate how well the disinfectant works, how long we have to use it for, and the dosage of it. So we can change the dilution, we can change the dose of it, we can dilute it more or add more, and we can test to see which ones kill more microorganisms. Okay, let's move on to commercial disinfectants. We have gas sterilization. So here we can use gas chambers to sterilize equipment. So this is very similar to what we see with autoclaving. We're putting something in a big box and treating it. So autoclaving deals with temperature and pressure. Here with gas sterilization, we're using a chemical like ethylene oxide, ETO, in order to kill off microorganisms. And this actually sterilizes them too. So we have gamma radiation that we can use here too, and antimicrobial gases. So they can be combined for sterilization procedures. Looking at antibiotics, we know antibiotics work well because they have what we call selective toxicity, meaning antibiotics target microorganisms. So they don't harm our tissues within our body. So our eukaryotic cells are not part of the mode of action of these chemotherapeutic agents, of these antibiotics themselves. When we use the term antibiotics versus chemotherapeutic agents is actually the larger category because we initially derived antibiotics from microorganisms, and those were natural antibiotics. But now we're synthesizing them and compounding novel antibiotics. And so we can call the whole category chemotherapeutic agents here. Antibiotics are kind of known as the magic bullet because, again, it only affects the bacteria, and that's what makes it selectively toxic. If we look back at viruses, we know viruses are difficult to kill because of the selective toxicity. Viruses use their own machinery, so it's very, very hard to target viruses. If we were looking at chemotherapy, treatment for cancer, that's even harder because here we're treating our cells in our body. We're trying to differentiate between the cancerous cells and the healthy cells. So getting something that's selectively toxic against the cancer cells when they're actually our own cells, is really difficult. So viruses are difficult again for antivirals, but bacteria is a lot easier because their cell type is so different. There's many different targets that we can use. Here on the right, we see an image of a child with a bacterial infection. Here we see the wound where the bacterial infection is spreading from the neck to the face. And then we can see the treatment with antibiotics. Before antibiotics emerged, children were dying from bacterial infections continuously. So this was a very, very normal thing to succumb to bacterial infections. If you look back at family trees hundreds of years ago, you would see typically children that were born and lost their life mostly to bacterial infection. So we've really increased our population by being able to control bacterial infections with these antibiotics and chemotherapeutic agents. Let's go ahead and look at how we can test which antibiotics are the most effective antibiotics. So we call this the Kirby-Bauer test, and it's what we saw on the initial slide of this PowerPoint. Here we can see that we have a Petri dish, and what we see, this cream color here is bacteria all over the Petri dish. So we'd start by spreading the microorganism continuously over the plate. Then we add these discs, these white little, they look like hole punch discs, which have antibiotic on them. So they're actually impregnated with antibiotic. And when placed on the plate, and then we left the plate to grow, we see that the bacteria cannot grow around some of them. So here we see this large clearing. This clearing is called a zone of inhibition. So very similar to the plaque assay we saw with the viruses. We saw a clearing because the virus lysed open the cells. Well, here, bacteria can't grow in this area. Anyone that gets close is typically lysed open or dies. So those clearings are how we can tell if an antibiotic is working, by how large the zone of inhibition is, and we can compare it back to standards. So looking at some of these antibiotics here, like the OX1 or this ERP, we see a very small zone of inhibition. This would not be the ideal antibiotic to use for this specific infection. So if this bacteria was specifically taken from a person with an infection and we wanted to know which antibiotic to use, this is how we could personalize and try which antibiotic is going to be the most effective against that specific type of bacteria. So typically the ones with the larger zones of inhibition are the ones that we use here. But again, it's compared back to standards. Okay, let's talk about antibiotics in clinical use. Here we can see samples, and we see these ones that are more foggy looking. We call that actually turbid. So what we're looking at there is the microbial growth inside of the tube that's suspended. Over here, we see these samples are more clear. The clearer the sample is, that means the less microbial microbes that are in the sample. So we go from a high concentration to a lower concentration here. And so we're increasing so we're decreasing the amount of microbes, but actually what's going on here, if we're looking at the bottom, we're increasing the amount of antibiotic. So if we treat a sample with 2 microgram per milliliter, we see that it is not effective at killing microorganisms. 4 microgram per milliliter, not effective. And then we see the edge case here, we reach the threshold. So we double it again from 2 to 4 to 8. And at 8, we see we have killed the microorganisms, and anything higher, like 16 micrograms, 32 micrograms, we have the same result. So this is how we can figure out the therapeutic dose that we need. So we wouldn't need 32 micrograms per milliliter because we already have killed off microorganisms at 8 microgram per milliliter. This is important because while antibiotics are selectively toxic and only target microorganisms, we still have to process them through our body, meaning that they're moving through our kidneys or moving through our liver and getting processed. And all of these additive chemical compounds within our body can alter our kidney function, can alter our liver, our liver cellular rates as well. So we don't want to give high doses that can have these alternative impacts that are less desirable. So that's why looking at the correct concentration is really important. What's the therapeutic dose, the minimum dose that we need for somebody's body weight to stop the growth? Interesting fact with the therapeutic dose, we have to look at the weight. So if we're looking at a child, maybe that's 50 pounds, maybe versus an adult, that's 150 pounds. We're going to change the therapeutic dose. You wouldn't use an adult concentration antibiotic dose on a child. So we have different dosages based off of weight. Looking at toxic dose, this is going to be important. That's the maximum dose that can be tolerated by a person. So again, if we gave the amount for a adult, an antibiotic for adult to a child, we might be surpassing the toxic dose, and it might become toxic to the child themselves and harm them. When we look at the chemotherapeutic index, we're looking at toxic dose to therapeutic dose. So we're looking at that ratio of how much can we give them without harming them. This is important because many of the antibiotics that are found are great and they kill microorganisms, but unfortunately, their chemotherapeutic index is not good, meaning that their therapeutic toxic dose and toxic dose are right next to one another. And when they're so close to one another, we see that the additive benefit of the antibiotic may actually come with additional problems because of the toxic dose of it is so close. So it's essentially toxic to take the antibiotic. So many antibiotics never make it to market because their toxic dose is too high compared to the therapeutic dose. Okay, let's talk about the use of antibiotics together. So if you just go to your general care practitioner because you're sick, you get an antibiotic, you typically just get one. But if you end up in the emergency room in a critical situation in which you need emergency IV antibiotics, they typically give you more than one antibiotic. But in order to determine which antibiotic to give a patient, we have to understand how these antibiotics work and whether they work in a synergistic fashion or an antagonistic fashion. So synergistic means that when we combine antibiotics like glycosides and vancomycin, they actually had an additive effect. So we could treat separately with them. Versus together, we see a beneficial effect. So maybe they have two different modes of action in how they work. One's preventing DNA synthesis. The other one's maybe preventing peptidoglycan synthesis or protein synthesis or metabolic reagents from being made within the body. So, or within the cell, I should say. So synergistic effects are pretty important, and we can combine drugs to get a mutually beneficial situation. Also, with synergistic treatment, we can see that just giving someone one drug, they may end up becoming antibiotic resistant, the microorganism. So by giving two at a time or a cocktail of drugs, it's less likely for someone to become antibiotic resistant. On the other hand, we have the antagonistic effect. So we have to look at the drugs that we're combining, like penicillin and macrolides. We want to make sure that they're not working against one another. So if one is knocking out the other or they're not effective together, it can actually be antagonistic. So we have to study synergistic effects by using a cocktail of drugs. When we talk about how antibiotics work, again, it's that selective toxicity. So we're targeting things that microorganisms do that our cells don't do. So cell wall synthesis, we have the peptidoglycan. Peptidoglycan is found in bacterial cells, not in our own cells. So it's a perfect target for selective toxicity. Cell membrane integrity, some of the membranes we can alter with these antibacterial and antifungal mechanisms, which we'll talk about later. Mycolic acid synthesis, we'll discuss DNA replication, transcription, protein synthesis, and metabolic reactions as well. So this is a good table to just kind of go through what we're going to find on these upcoming slides. If you're a visual person, this slide may be better to help understand how antibiotics can work. And again, they have a different mode of action depending on which one we're looking at. So here we see peptidoglycan, which is again used to bacterial cells. It's unique to them. And we can use cell wall inhibitors to break down or prevent the growth of peptidoglycan. So as the cell grows and divides, it needs to replicate and produce more peptidoglycan. If we prevent that from happening, the cell can lyse open. So we can kill microorganisms this way. So we have all of our cell wall inhibitors, and probably ones you've heard of, like penicillin, vancomycin, cephalosporins. Those all work on peptidoglycan. It's a very common mode of action, and we know penicillins were first antibiotics found by Alexander Fleming on accident. Okay. When we talk about antibiotics, antibiotics are actually produced mostly from bacterial cells towards other bacterial cells. So while penicillin was found in Penicillium, a fungus that was killing off bacteria, bacteria and bacteria fight one another. Remember, they're living in these extremely diverse environments with other species of microorganisms, and they're fighting for resources, for territory to live in. So they're producing antibiotics to fight one another. Okay. Here we also have parts of the central dogma that can be affected. So that's going from DNA to RNA to protein. Here we see DNA replication with our quinolones, RNA inhibitors, and then even down to protein synthesis, whether we're hitting the 50S or 30S portions of the ribosome. So DNA replication can prevent cells from dividing. RNA, preventing RNA from preventing transcription from correctly functioning is going to prevent us from being able to effectively make proteins or make RNA products. And protein synthesis, we know proteins are the most ubiquitous macromolecule within the body. So if we cut off protein synthesis, we cut off really a lifeline for the cell because we lose all of these different types of functions within the cell. Not only do we have that, we have cell membrane damage. So if we think of that phospholipid bilayer, we can actually break it down. So we'll look into that in just a bit. And then we have metabolic inhibitors. So all the metabolic reactions that allow the cell to survive, we can prevent some of them from occurring that are different from our metabolic reactions. Okay, looking at peptidoglycan, here we can see our NAG and NAM assembly with our amino acid linkage. So here we see the production of NAG and NAM. So NAG and NAM are made inside of the cell, and it's actually flipped across the cell. We have flippase flipping it across the cell, and it becomes assembled on the outside of the cell. We can use specific antibiotics, which can break down the attachment sites that we see here. And remember, it's like a chain link fence that holds the cell together and helps with osmotic regulation of water going in and water going out. If we break open that chain link, all of a sudden the cell is susceptible to osmotic irregularities, which can cause lysis of the cell. So here we see penicillin, vancomycin, cephalosporins are all going to alter the way that it actually connects together, where bactracin and bactracin is actually working on preventing the monomers of peptidoglycan, the NAG and NAM, from moving outside of the cell. Okay. Looking at most antibiotics that work on peptidoglycan, we call them beta-lactam antibiotics. So these are beta-lactam antibiotics. They use a beta-lactam ring. So here's our beta-lactam ring. And what can actually happen is that we can get antibiotic resistance to this beta-lactam ring. So we have microorganisms that produce penicillin binding proteins, or PBPs. Penicillin binding proteins bind to antibiotics such as penicillin and alter this beta-lactam ring. If the beta-lactam ring is a functionally active chemical portion of the molecule, well, if we alter that, it's no longer functional. So microorganisms have adapted to become antibiotic resistant, specifically against any antibiotic with the beta-lactam ring. And that's what we're seeing here. These ones use beta-lactam rings for their functionality. Okay. So what we've done is when microorganisms become resistant to antibiotics, we alter antibiotics. And this is where we start modifying and synthesizing new antibiotics where this beta-lactam ring can't be modified. And then, of course, what we can imagine happens is that bacteria adapt and evolve to be able to fight off even the newest changes. So we're constantly at war with bacteria. So we throw one thing at them, like antibiotics with a beta-lactam ring. They go ahead and evolve, and then they can spread around those genes with horizontal gene transfer. So then we have to throw a different antibiotic at them. Then they evolve again. Then we have to throw another antibiotic, and then they evolve again. So we're constantly at warfare with microorganisms, and antibiotic resistance is something that we're continuously facing more and more of as we're giving them more exposure to antibiotics. So antibiotic resistance, again, with the beta-lactam ring, here we can see beta-lactam ring can be altered by beta-lactamase. Beta-lactamase is going to break that beta-lactam ring, and we lose the functionality of the beta-lactam ring when it changes conformation, when it changes its molecular structure here. So the ability for microorganisms to be antibiotic resistant falls on horizontal gene transfer or mutation. So with horizontal gene transfer, we have conjugation, microorganisms connecting to one another, cell-to-cell contact, cell sex through a pilus, exchanging genetic information. We have transduction, which is using a bacteriophage, which can bring in genetic material from one cell to the next. And we have transformation, microorganisms picking up exogenous DNA in the environment around them. Well, that exogenous DNA can be a gene that confers for antibiotic resistance, just like we see here. So microorganisms are constantly evolving and they're getting new genes through mutation. Every time a cell divides is an opportunity for a mutation to take place that then can be passed down in vertical transmission or horizontal transmission. So they're very good at adapting to their environment. Okay. Let's go ahead and look at different types of drugs here. We have polymyxins. Polymyxins can alter the cell membrane integrity. Here we see our phospholipid bilayer, our heads that are hydrophilic, our tails that are hydrophobic, making them amphipathic, both hydrophilic and hydrophobic at the same time. The membrane with this structure has the hydrophobic tails interacting with the hydrophilic heads interacting with the aquatic environment around them. This structure is built with the tails together due to the amphipathic nature of phospholipids themselves, and we create the phospholipid bilayer. Phospholipid bilayer is the original formation of outside environment and inside environment. So for the first cell to exist, this is what separates the outside world from the inside world. And this is a really important distinction because cells have to keep their internal environment regulated. So they run their metabolic reactions, they regulate the salt, the pH, based off their internal environment. So this phospholipid bilayer is selectively permeable. It lets in certain things, but not other things. So if we alter the functionality of the selectively permeable membrane by poking holes in the cytoplasmic membrane, we can essentially kill the cell. So here we see a pore essentially being drilled through the cell. This is a hydrophobic side chain. It's interacting with those hydrophobic tails. So we drill these pores through and it can essentially lyse open the cell. We also have detergents. So looking at like polymyxin, detergents are going to alter membrane integrity. So just like if you're washing grease off of your dishes, you're using a detergent, right? So when we have oils on our plate before we put it in the washing machine, we use a detergent to remove those oils because it alters this hydrophobic interaction of the way that the tails are holding one another together. We also have some other antibiotics that prevent fatty acid synthesis because that's what these lipids are. Lipids are fat. So if we prevent their synthesis, then we don't make more lipids. And as a cell divides, it's going to need more lipids for the membrane. Okay. Moving forward, let's look at DNA synthesis. So we'll cut into the central dogma. Central dogma has two functions. DNA synthesis is for cellular replication. We have to double that DNA before we divide it in binary fission. Then we'll get to gene expression with transcription and translation following that. Quinolones are the category of drugs that alter DNA synthesis. Quinolones can work on the DNA gyrase. So gyrase is an enzyme that's found upstream of the DNA as it's dividing. So as the DNA is opening up, so the bases are exposed, we can start replicating new bases on them. The DNA has to be constantly unwound. And DNA gyrase actually does this. It starts removing those coils and supercoiling as we're unraveling the DNA to read the bases. So quinolones block DNA synthesis from preventing DNA gyrase from functioning. So it can't fix that supercoiling and it can't open up the DNA double helix to go ahead and replicate it. So we've effectively stopped DNA replication, which then affects cells from actually dividing again. Let's look at protein synthesis. So here we're targeting the ribosome itself. The ribosome has two components. It has a 50S subunit, the larger one on top, which is a sedimentation rate. and the 30S subunit found at the bottom. We have specific antibiotics that work on the 50S, specific ones that work on the 30S, like tetracycline, aminoglycosides, or macrolides on the 50S. All of these are going to prevent protein synthesis. And again, proteins are critical within the cell. They can be part of the membrane for receptors. We have proteins that are enzymes that run critical metabolic reactions. We have proteins that work with DNA. So protein synthesis inhibitors are extremely important in the category of antibiotics. We also use metabolism inhibitors like these sulfa drugs or sulfamides that we see here. These can prevent the synthesis of folic acid. Folic acid is necessary for the production of DNA and for the production of RNA. So looking at the central dogma, we can't divide cells because if we can't make DNA, we can't replicate our DNA. If we can't make RNA, we can't start transcription. So folic acid is extremely important, whether we're looking at gene expression, which then long-term affects protein synthesis, or whether we're affecting binary fission and cells even dividing. So let's go ahead and look at how PABA creates folic acid. So PABA is our aminobenzoic acid, PABA, and then we have our SFA here. So we're going to see this interaction. So normal function of PABA, it works with an enzyme and it connects these terminal ends and we make folic acid. So the enzyme brings together these three portions and generates folic acid for a healthy functioning metabolic cell. But on the other hand, if we use these SFAs, they're going to interfere with the way that PABA is actually synthesized into folic acid. So they look analogous. SFA, the molecular structure of it is very similar to PABA. So SFA is grabbed by the enzyme instead of PABA, and it grabs those terminal ends, the P and G that we see here, and it grabs SFA, and it glues SFA and G together. So now we don't have available G to glue to PABA because the enzyme is mistaking this antibiotic for PABA itself, the para-aminobenzoic acid, which is a critical component of folic acid. So these sulfonamides are important in preventing metabolic reactions from occurring. So it's tricking these cells into incorporating the antibiotic instead of incorporating PABA in this metabolic reaction. Okay, let's look at antibiotic resistance and what can happen. There's a lot of information in this picture, so we'll go ahead and walk through it and give you a minute to look at it as well. So here we can see mechanisms that are preventing accumulation of antibiotics. So some antibiotics, like the penicillins we see, target the cell wall on the outside, the peptidoglycan, but some of them have to work their way into the cell, and they're chemically active in the cell. So if they're active on the DNA, if they're active on the ribosome, if they're active in metabolism, they're mostly in the cell. So quite a few of these antibiotics that we're using actually have to get into the cell. What we see happening here is that we have things like efflux pumps. So even if the antibiotic gets into the cell, it can be pumped right out of the cell. So instead of building up a high concentration of these antibiotics, it's pumped out of the cell. So you can imagine if SFA is an antibiotic being brought into the cell and we have an efflux pump pumping it out, it's no longer effective and PABA is going to be in competition with it as it naturally does. So we can pump out antibiotics. We can also destroy antibiotics. We can break them down or alter them. So like the beta-lactam drugs can be altered. So we're changing their chemical structure, which in turn changes their function. We can also change membrane permeability. We know the membrane is selectively permeable, so some antibiotics can go in, some cannot. Well, by altering membrane permeability, we can prevent specific antibiotics from entering the cell. It's a great defense mechanism and makes these cells antibiotic resistant. Looking at preventing binding, so we could alter the site. So specifically looking at protein synthesis, we know that some of them are going to interact with the 50S subunit, some with the 30S subunit. Well, if we block the point where these antibiotics interact with the ribosome or with the process of translation, well, then the antibiotic no longer works. And that's what we see here. So we see a mutated version of the ribosome. Now the antibiotic has lost its binding site and can no longer be effective in preventing protein synthesis. We also have mechanisms that can dislodge antibiotics. So here we see our ribosome, and we see the ribosome protective protein. So here we have macrolide trying to alter the ribosome, which we saw back here. Macrolides work on the 50S subunit here. It's preventing the peptidyltransferase, so bonding of the two amino acids together here. So what we can do is we can have proteins that are generated in the cell. So these can be things that came about from mutation, or we've received them in horizontal gene transfer. Bacterial cells have, and they can work to push out antibiotics. They can dislodge the antibiotic from the ribosome, restoring the functionality of the ribosome. Let's look at drug resistance in general and what can happen. So this image, I think, is a really good kind of simplified way of looking at it. Here we have a patient's lung, and we see that we have bacteria within our lungs. What's going to happen is the bacteria is going to start replicating. It's colonizing the lung, dividing over and over through binary fission. And in the process of division, every now and then a mutation comes about as we replicate the DNA. So here we see spontaneous antibiotic resistant mutation in red. Okay, so we go ahead and take the antibiotics. When we take the antibiotics, it kills off all of the other microorganisms. So all of those blue ones that are susceptible to the antibiotic die off. But our one that is not susceptible has now replicated. It's undergone binary fission, and it's now 2 cells instead of 1. So what we can see is that the patient starts feeling better. They start feeling like they're recovering. But all of a sudden, this pneumonia comes back. So this bacteria goes from 1 cell to 2 cells to complete replication logarithmic cycle. And now we have an antibiotic resistant strain within the lungs. We can no longer treat with the same antibiotic. Step 2 of using it again will not work. So we have a relapse in our signs and symptoms for the patient's illness and deterioration of function. So I think this is a really good example for understanding antibiotic resistance. Okay, let's look at viruses. We talked about some ways that we have antivirals. Again, antivirals are more difficult to generate because viruses rely on our host machinery, and that's part of why they're non-living. So if they're using our DNA synthesis, if they're using our RNA polymerase, if they're using our ribosomes, then they're dependent really on our cells. So then we can't target a lot of those mechanisms because we'd be targeting our own cells' mechanisms and preventing our own cells from being viable themselves. So some of the unique things we can look at is membrane coating of the viruses. So looking at viruses that impact humans, we know a lot of times they bud out of the cell. As they bud out of the cell and take the membrane with them, we can prevent that activity. So membrane coating of the viruses, we have like Tamiflu, which again is a neuraminidase inhibitor that prevents the viral particle from actually leaving the cell. So the viron gets cross-linked at the edge of the cell and cannot actually be exported out of the cell, so it can't go on to affect other cells around it. Here we see uncoating is another thing. So as the virus comes into the cell, we have to remove those outer layers of the virus. And this typically happens in the endosome. So the endosome changes pH, and this allows the release of the virus. Well, we can alter this process with an antiviral medication that prevents uncoating. Looking at viral DNA synthesis, there are some antiviral drugs and RNA synthesis inhibitors that can be used. So this can affect DNA synthesis and can affect protein synthesis, or RNA synthesis, which then inhibits protein synthesis here. So some of these drugs can inhibit viral DNA synthesis. So it's unique structures that prevent this. So here we have acyclovir is a good example of one. Acyclovir is used for herpes simplex virus, so it will prevent replication of the virus. Okay. With HIV, we talked about protease inhibitors, so ones that are breaking down proteins. We can also prevent them from entering into the cell. So here's our CCR5, one of the receptors on the outside of the cell, kind of like the CD4, but CCR5 receptor that interacts with HIV. If we block the receptor, then the virus cannot interact with the receptor, it won't have that attachment, and be able to move into the cell. So we have what we call ART, which is antiretroviral therapy. Antiretroviral therapy has even changed to the point where we call it HAART at this point, because we have highly active antiviral retrotherapy for HIV infections today, which again is saved many lives and reclassified an HIV infection to more of a chronic disease than a disease where we see high rates of fatalities. We also have antifungal agents. So not only do we have antivirals, antibacterials, we have antiprotozoa drugs, and we have antifungals. We're not going to go through this whole list, but remember, we have to find something selectively toxic. So we have to find something that's different about fungus than our own cells. So here we can see we have some antifungals that block fungal cell wall synthesis. We have chitin in the cell wall. Chitin is unique to fungus in the way that peptidoglycan is unique for the cell wall of bacteria, or like cellulose for plant cells, unique cell wall structures. These are the things we can go after, anything different about the cell. Okay. Let's go ahead and move on to the questions. We don't go into too much detail with antifungals, just know that they're existing. Let's talk about question number 1. It says, complete destruction of all microorganisms on a surface can be achieved by. So again, we'll give you a minute to read over this and pause. And the correct answer would be sterilization. So a couple keys in here that we can look at is on a surface. So a surface, we're not treating a human body. So on the surface, we can sterilize. And if we have complete destruction of all microorganisms, we're talking about viruses, we're talking about endospores, all bacteria, that is sterilization. That's the peak of killing microorganisms. Oh, did I miss question? Oh, we go on to question number 3. Hypothesize why there are more chemotherapeutic agents that block bacterial protein synthesis than chemotherapeutic agents that block viral protein synthesis. Again, we'll give you a minute to look over this and pause the video. Okay, moving on. Question number 3. The correct answer is that viral protein synthesis relies entirely on host ribosomes. So viruses don't bring in their own ribosomes into the cells. They're using host machinery, one of the reasons why they're not alive. So we have more drugs that can target bacterial protein synthesis because bacteria use their own proteins, where on the other hand, viruses use our proteins, and we don't want to inhibit our protein synthesis machinery from working. Question number 4. Which of the following is a target for an antiviral drug? I'll let you read through the answers and pause the video. Okay, here we see DNA and RNA synthesis are targets for antiviral drugs, not the only target for antiviral drugs. We just talked about viruses uncoding and viruses budding out from cells being prevented. But this is a great example, DNA or RNA synthesis. Okay, here we've reached these section objectives. These are for you to review after the video and go back and fill out to make sure that you're getting the main points from the video. And that wraps up essentially everything we see here for this lecture.